Today we will have the overview of the lipids digestion and absorption. As we all know, lipids are fats and fats-like substances that store energy. They are insoluble and the lipid constitute about 15 to 20% of our body weight. The adult can ingest 0 to 150 milligrams per day of the lipids. There are two types of the sources of the lipids, which are the exogenous and endogenous sources. Exogenous sources we have tags, 90% tags, triacylglycerols, and the remaining 10% include cholesterol, cholesterol esters, phospholipids, and free fatty acids. The endogenous sources includes the lipids which are synthesized in our body cells. First, we will have the digestion in the stomach. There are two enzymes, lingual lipase and gastric lipase, which targets the short-chain triacylglycerols. These enzymes are acidic stable, means their pH is stable at 4 to 6. These enzymes hydrolyze the milk fats in the neonates, also in the individual with pancreatic insufficiency. Secondly, emulsification in the small intestine. By emulsification, we mean the breakdown of the large fat globules into a tiny droplets by reducing their surface tension. The purpose of this is to increase the surface area for enzymatic actions. This is the fat globule, large fat globule, which is emulsified to form the tiny droplets, and these blacks are the bile salts. That means the tiny droplets are surrounded by the bile salts. There are three mechanisms in the emulsification: the detergent action, surfactant action, and peristalsis. Detergent actions, bile salts, the biological detergents derived from the cholesterols in liver. They stabilize the tiny drops by prevention from the coalescing. In bile salts, we have the steroid nucleus and the side chain. The surfactant action. In emulsification, the equation is lipid which emulsify to form the free fatty acids and the monoacylglycerols. For the surfactant, the monoacylglycerols and the free fatty acids combine with the phospholipids, which is the lecithin, to form the surfactant. This boosts the emulsification. Then we have the peristalsis, mechanical mixing, which also aid in the digestion. Then there is a digestion by the pancreatic enzymes. The fatty acids in the presence of the pancreatic lipase will form the two monoglycerides and the free fatty acids. Basically, this enzyme, pancreatic lipase, acts at the position 1 or 3, and as a result, we will have the two monoglyceride. The pancreatic lipase is inhibited by the bile acids, to which another enzyme, colipase, come and protect the lipase. Then we have the cholesterol esters, which in the presence of the pancreatic esterase will give you the free fatty acids. The fatty acids in the presence of the phospholipase A2 will give the free fatty acids and lysophospholipids. Moving on to the absorption of the lipids. For the absorption, we have many theories. The most important theories are lipolytic theory, partition theory, or the Bergstrom theory. The first theory indicates that the fats completely hydrolyze to the glycerols and the free fatty acids. The partition theory indicates that the tags digestion is partial. The most recent and advanced theory that we are following is the Bergstrom theory. according to which the triacylglycerols in the presence of the pancreatic lipase will form the two monoglycerides and the free fatty acids. The mechanism of the absorption is the formation of the micelles, which are from the bile salts. They are disc shaped and the bile salts at the periphery. In the center, we have the monoacylglycerols, cholesterols, esters and other compounds, and it is surrounded by the bile salts. The micelles transport lipids from the lumen into the enterocytes, which is the site of the absorption. In the enterocyte, we will have the chylomicrons, which then transport the lipids from the enterocytes to the circulation, first into the lymph and then to the blood circulation. For the structure of the chylomicrons. The chylomicrons are outerly covered by the amphipathic layer and inner by the hydrophobic. On the outside, we have the apolipoproteins, and inside we have the tags and cholesterol esters.